Simon LeVay got everyone's attention well before we did when he looked at a brain region in the hypothalamus, a specific region called the preoptic area, or POA, and he looked there and compared the size of the POA in the brains of gay men versus straight men. And he looked in the preoptic area because in rats, there's a very prominent sexual difference or sex dimorphism in a nucleus in the rat brain in the preoptic area. And the nucleus got named the sexually dimorphic nucleus of the preoptic area, the SDNPOA. And Simon knew there was a huge sex difference there in rats. And so he looked at the brains of gay and straight men and found a nucleus there that may or may not be the same as the SDNPOA, but is larger in men than in women. And what he found was that the nucleus in gay men was smaller than in straight men. And in fact, not significantly different from the size of the nucleus in women.
So it wasn't hyper male like the finger length ratio was?
No. In that case, it indicated either less androgen exposure or less of a response to the androgen that was there. And Simon got even more of an uproar than I did, published his paper in Science, and there were lots of people that were very skeptical, including some neuroscientists, but eventually another group replicated it.
That's what I was going to ask, because I recall that the two major critiques of the paper, one was fair in my opinion. It was that some of the postmortem samples were from people who had died of AIDS, and so AIDS has some known neurodegenerative effects that may or may not have impacted the samples, although hopefully they control for that. And then as I recall, he also got some pushback because he is openly gay, and people accused him of agended. I didn't say gendered. I said agended science.
Yeah, that he was part of some conspiracy, a gay agenda to force Americans to regard people with same-sex orientation as somehow okay.
Mm-hmm. But a replication of the study from an independent group that presumably has no reason to be biased whatsoever.
Well, they were very skeptical of it. So that was William Blyer, who eventually there's an interesting aspect of it. It took him a long time to get a sample big enough because, Simon, it was such a horrible time in AIDS epidemic. There were so many young men dying that Simon had no trouble finding enough brains to do the sample. And then as treatment got better, the death rates of HIV started going down. And so it took longer for William Blyer to gather the samples, but eventually did. And even though he was skeptical of it, he saw it too. The question about AIDS, I mean, Simon was able to address that in that he also had some straight men who had AIDS, and he didn't, they weren't significantly different from other straight men. And so it was widely interpreted as proof that sexual orientation is not a choice, that it's something that happens to you. And of course, I don't think sexual orientation is a choice. That's true. But Simon himself made it clear that he could only look at this nucleus in adults, right? You can only look, it's so tiny. The sexually dimorphic nucleus, the preoptic area in humans, it's about the size of a grain of sand, right? So you got to have a microscope, and you can only, there's no non-invasive way to look at it. And he pointed out that he didn't know what the order of causation was. He didn't know if those men had been born with a smaller SDNPOA, and that's why they became gay, or did something else cause them to become gay and also cause the SDNPOA to get smaller. And for the public, the idea that a nucleus might change its size in adulthood, maybe that seems kind of like, unlikely. But as neuroscientists, we know, that adult brains are changing all the time. In fact, even in animals, Brad Cook showed that, there's a nucleus, the medial amygdala, there's sex difference there, but if you take away the testosterone in males, sex difference goes away in just a matter of a few weeks. So Simon's work on the SDNPOA, also known as INA3, I won't bother with why.
Unfortunately, I remember the full name because my brain, it's the interstitial nucleus of the anterior hypothalamus subregion 3. Yeah. I'm not saying that to impress anyone. When you get to be 50, which I can now say that, you wonder why your hippocampus remember certain things that are like basically totally useless to remember.
I waste so many synapses on totally useless crap, but that's worth holding on to. You did well. So so we don't know. It's a chicken and egg problem. We don't know if that happened, if they were gay because they had a small I to 3, or do they have a small I to 3 because they're gay. So what I liked about Dennis's otoacoustic emissions is pretty good evidence that happened well before they had a sexual orientation. And the other thing I always liked about it is that, well, I mean, you can't imagine there's a social influence on digital, and nobody knew about this. So I wasn't worried that there were some little girls out there that looked at their hand and say, gee, that looks kind of masculine. Maybe I should be a lesbian, right? Because nobody knew this.
Until your paper was published.
Yes, yeah.
I mean, as I recall, there was some like schoolyard stuff of kids looking at each other's hands and trying to decide who was gay and who was straight. And
every once in a while, to this day on the internet, I'll look at it and there'll be a little ad over there claiming to tell me something about my personality based on digit ratio length, and it's just like, please, there's nothing to it. Don't waste your money, gang. And there's an aspect of this fact that the group differences were there, but you can't tell about differences between individuals, that I think is the hardest thing for scientists to communicate to the public at large.
You have a really good way of explaining this to people and it's an important lesson just in reading statistics and making sense of data that I'd love for you to give an example of how this plays out, perhaps in a separate example.
Psychologists, they like to talk about a way of measuring how big a difference is between two groups. So this difference in digit ratios between men and women, it's a relatively small difference. It's like, well, we measure it in terms of how many standard deviations apart are the two means of the populations.
The two averages.
The two averages. And to give an example of that, everyone knows about the sex difference in human height, right, among adults. So that's a huge sex difference. It's one where we're all aware of it, and if nobody had told us there was such a thing, we'd notice it after a while, right? And that's because those two averages and those two populations are about 2 standard deviations apart, the standard deviation being a measure of how much variability there is in something. So that's a huge sex difference. And to give you an idea of what that means, if I had you grab a sample of 1,000 people, and I'm going to tell you one thing about each one of them, and that's their height. That's all. And now I give you the job. You've got to guess what sex they are. And I can tell you what you want to do to maximize your hit rate. Everyone who is 5 feet, 5.5, you're going to say those are men. And if they're less than that, you're going to say they're women. And you'll be right about 80% of the time, which means you'll be wrong almost 20% of the time, right? And so everyone, you can see that there's some predictive power there, but it's far from perfect. Well, this sex difference in digit ratios is half a standard deviation, so a quarter of that. And so that means there's much more overlap. And we know that other things influence digit ratios too, not just prenatal testosterone. And so this is why there's no predictive value. I mean, so anyone who looked at their hand while we were talking about this and got worried, I don't know anything about your particular prenatal testosterone level, no matter what your digit ratio is.
What about bisexuality? People who identify as attracted to both men and women, and maybe I ask about first bisexual women, then bisexual men. Is there a pattern in digit ratios that leads anywhere?
I mean, in those days, we didn't have enough people that identified as bisexual to have a reasonable sample. And it's interesting, and that's something that's changed, right? I mean, if you do surveys now, especially among younger people, there are more people who report that they are bisexual than there were then. So I don't really have anything to say about them. The one thing I will say is I'm sure that even among lesbians, there's more than one pathway, more than one developmental pathway to become a lesbian or to become a gay man, right? I don't think there's just one thing. That's not how human behavior.
But you do think it's based on the data that it's biological?
Based on that data, I think testosterone has a say, right? That it doesn't mean it's the whole package.
Prenatal testosterone.
Prenatal testosterone.
And the reason I ask that is, I mean, there are conditions that are not uncommon where someone has a particularly stressful, long phase of development where there's every reason to believe that their androgens are impacted negatively.
Yes.
There's also every reason to believe that there are stretches of development where androgens are increased. Like we know that people who do a sort of a contact sport
Right.
or engage in anything that requires like deliberate aggression. I realize that real martial artists know competitive aggression, know how to, sort of gate their levels of aggression so that they're not like in a fury, right? But we know that certain types of activities, competition, et cetera, I mean, there's no question that those can increase androgens. So you can imagine there's some plasticity postnatally, and that could be before puberty. It could be during puberty. And so that's testosterone. The body, as you said something that even as a developmental neurobiologist, I don't think I'd ever heard stated so clearly. And it's so important that the rate of brain development from birth until age 12 is at least as fast as it was before we're born.
Yeah, the way to really bring that home is to compare human brain growth and chimpanzee brain growth. So up until birth, the rate at which the brain size increases compared to body size is about the same in humans and chimps. And shortly after birth, the chimpanzee brain stops growing as fast and eventually asymptotes right away. The human brain continues that feverish fetal rate of growth until at least 6 years of age, maybe out there to 10 years of age. Mm-hmm. So people have pointed out that in a real sense, human beings, children, are fetuses that are outside learning a whole bunch of stuff from other people. That's the real distinctiveness of our species is, we have this protracted childhood and really intense social learning, and as you say, a fetal rate of growth, even though we're not in the fetus anymore.
So I guess for me, the idea that behavior, exposure to things, and I don't want to get into valence of negative, positive, but sure, pesticides, but also, schoolyard activities.
Yep.
If you have, and we'll get to this, 5 siblings and it's very competitive, who gets how much pizza. I've been, I had one sibling, so it was a little bit, there was competition, but it was different, right? Yeah. These things change hormones. Hormones change the brain. The brain, as you're explaining, can impact sexual preference.
What's great about studying hormones and behavior, right, is that sometimes you can control the hormone, in animals at least. But the hard thing about hormones and behavior that people don't understand is that behavior can affect hormones, as you say. In competitions, the winners afterwards are more likely to have higher testosterone and the losers will have lower. In elections, it's been shown that people whose candidate won the presidential election, their testosterone levels went up a little bit, and the people whose candidate lost went down a little. And so you'll always have this cycle where the hormone alters the behavior, and then the behavior alters the hormone, and you always have to look for ways to try to pin down the order of effects, and not always easy. The only thing I know for sure is that the brain remains plastic all of our lives.
Well, that statement is a significant one because I also believe that the brain remains plastic throughout our lives. It's always surprising to me that the hypothalamus remains plastic throughout our lives. And I think that's worth perhaps double clicking on, so to speak, because the idea that you can learn another language and your neocortex changes, or you can learn to juggle and your motor cortex changes, your cerebellum changes. Okay, like there's a lot of beautiful studies demonstrating that. But when I think about the hypothalamus, I think about it as something that's pretty hardwired by time puberty wraps up. Mm. But the more I learn and the more data that get published, the more surprised I am. I saw a paper just the other day that the neurons that control suppression of appetite, these POMC neurons in the arcuate nucleus, there's a population of them that are sort of undifferentiated that can become, let's just call it pro-hunger. and by expressing a different peptide, neuropeptide Y, and that there's a lot of late stage plasticity. And this may explain why people who reach a certain level of obesity may actually find that they're hungrier despite not eating food. So it's fascinating to me how these deeper brain structures may actually remain plastic.
I think both your statements are true. I think it's probably true that the neocortex is more plastic than the hypothalamus, but it's a matter of relativity. And so, as you say, the one thing we know is that there's plenty of plasticity there. The other thing I noticed, so I've gone to Society for Neuroscience meetings pretty regularly every year since 1977. And after a while, I noticed something. Every year when I went to the neuroscience meeting, the brain was more plastic than it was the year before, right? I mean, because there are more and more of these demonstrations, and it's like, I think synapses can come and go just about anywhere, and so hardwired. Well, let's say might be less plasticity in the hypothalamus, surely so, but that doesn't mean there's none.
As many of you know, I've been taking AG1 for nearly 15 years now. I discovered it way back in 2012, long before I ever had a podcast, and I've been taking it every day since. The reason I started taking it, and the reason I still take it, is because AG1 is, to my knowledge, the highest quality and most comprehensive of the foundational nutritional supplements on the market. It combines vitamins, minerals, prebiotics, probiotics, and adaptogens into a single scoop that's easy to drink, and it tastes great. It's designed to support things like gut health, immune health, and overall energy. And it does so by helping to fill any gaps you might have in your daily nutrition. Now, of course, everyone should strive to eat nutritious whole foods. I certainly do that every day. But I'm often asked, if you could take just one supplement, what would that supplement be? And my answer is always AG1, because it has just been oh so critical to supporting all aspects of my physical health, mental health, and performance. I know this from my own experience with AG1, and I continually hear this from other people who use AG1 daily. If you would like to try AG1, you can go to drinkag1.com/Huberman to get a special offer. For a limited time, AG1 is giving away 6 free travel packs of AG1 and a bottle of vitamin D3K2 with your subscription. Again, that's drinkag1 with the numeral 1.com/Huberman to get 6 free travel packs and a bottle of vitamin D3K2 with your subscription. What are some of the other effects in human studies of behavior impacting hormones that come to mind for you?